talk is going to be about viruses. Viruses are acellular collections of biomolecules that infect cells. So what acellular means is they're not actually cells. Instead, there's going to be a collection of biomolecules, as we learned before, that have the ability to attach or get inside of what we call a host cell, a cell that it infects, and it changes the machinery of that cell forever. Let's take a look at the learning targets of what we're supposed to know by the end of today's lecture. First, explain whether a virus is technically alive or not. 2, describe the structure of a virus. 3, explain how a virus infects a cell. 4, compare and contrast lytic versus lysogenic viral cycles. 5, explain how a retrovirus works. And 6, define viroid and prion. Let's get to it. The viruses are typically associated with a number of plant, animal, and human diseases. We actually consider them non-living because they don't exhibit all of the criteria for life. Early in a biology class, you learn a whole list of characteristics that living things do. However, viruses are unable to acquire energy or utilize it by way of cellular respiration. They don't typically grow, nor do they develop more than their original structure. Let's look at some basics about viruses. They're extremely small, and as we said, they're non-cellular. They're not actually cells. Inside of them, they have a DNA or RNA genome. That genetic content is pretty small, from 4 genes to about 100 genes in size. And they can only reproduce using the machinery of a host cell. They can't actually copy themselves without hijacking the machinery inside of a cell that they infect. They don't have a typical scientific name, there's no genus and species, but instead they're often named scientifically for proteins sticking off the surface. Or even their common names can reflect the disease they cause or the organ and tissue they infect. For example, poliovirus causes polio. Tobacco mosaic virus infects a tobacco plant and it causes a mosaic of spots all over the leaves as it damages them. Here you can see a virus. It's very small next to the cell in which it's trying to infect. You'll notice that it has proteins that stick off of it. That's what these little barbs are all over the surface of this virus. And you can see there's other viruses nearby as well. This is a TEM, a transmission electron micrograph of a flu virus. You can see that it kind of has like a brush border on the outside. That's what those little proteins are that you see in this one here from the outside. And this right here, this is also a virus. This is called a bacteriophage. It's a virus that actually infects bacteria. So bacteria can actually get infected by viruses. Historically, viruses were detected indirectly long before they were actually seen. Tobacco plants have a common infection called tobacco mosaic disease, and it will stunt the growth of the tobacco plant and give their leaves almost a mosaic coloration of yellow spots on the green leaves that looks like this. The leaf on the right has been infected by the tobacco mosaic virus. Now, in the late 1800s, the researchers hypothesized that a particle smaller than a bacteria caused the disease. They actually couldn't find a bacteria that was responsible for it, so they had to go ahead and further test to see what they were dealing with. So what happened first was they extracted sap from a tobacco plant that had tobacco mosaic disease. And you can see right here, this leaf has those yellow spots. It was poured through a special tool, sap through a porcelain filter, that the pore size was small enough to trap any bacteria and allow the rest of the liquid sap to flow through, containing anything smaller than bacteria. They went ahead and they took the filtrate then and rubbed it on the leaves of healthy tobacco plants. This is called the Ivanowski experiment. What they found was that the healthy plants then became infected with tobacco mosaic disease, which ultimately led to the conclusion that if this trap here was collecting bacteria and not letting any pass through, it must be something smaller than bacteria that's causing tobacco plants to be infected. In 1935, Wendell Stanley confirmed this hypothesis by crystallizing the infectious particle now known as tobacco mosaic virus, or TMV. When the electron microscope was invented, these infectious agents, these virus particles, could now be seen, as we could never see something so small before. The virus structure itself, as we said, is very small, 20 to 40 nanometers. So we're talking about 20 billionths of a meter, which is incredibly tiny. Each type has at least two parts. First, the capsid. The capsid is the outer layer composed of proteins. Now, some viruses will have a membrane envelope on the outside of those capsids as well. However, both types will have nucleic acid cores, which means that it has genetic material, nucleic acid, DNA, or RNA inside of each viral particle. The overall shape can vary from thread-like rods to polyhedrals to complex space probe type shapes. Here we see a thread-like rod. You can see the proteins wrap around almost like an old school telephone cord, and on the inside you see an RNA core. This one here would be our space probe. This is common in viruses that infect bacteria. This one here, this polyhedral, is called an icosahedral. It has 20 sides and protein spikes that stick off of it, which causes human influenza or the flu. So here we'll look at a general breakdown of the structure of viruses. Virus particles have a covering made of a capsid, which is ultimately made of protein, and it can have an envelope. So not all viruses do have envelopes, but some do. The inner core of a virus particle, as we said, is nucleic acid. It's its genetic material, and it can be DNA or RNA, and it can also carry some proteins in the form of enzymes with them as well. Here's a closer look at some examples of different viruses. This is the tobacco mosaic virus. You can see that it has its capsid in a cylindrical type of shape with those protein spikes sticking off of all sides. You can see that the RNA also coils on the inside. This is a plant virus. The adenoviruses, they are typically your cold viruses, and they will infect typically your upper respiratory systems. They are considered a naked virus in the fact they don't have an envelope. They do have essentially glycoprotein coats, and they have protein spikes that stick off them, which we'll explain in a little while. Inside you can find DNA. This one here is an icosahedral shape. It has 20 triangular faces on its outer surface. Influenza viruses, which cause the flu, they are going to be coated in that membranous envelope we were talking about. It's going to help them sneak into cells. They typically are going to have an RNA core. They have protein capsids around those RNA cores, and then this membranous envelope on the outside with the protein spikes exists, and we'll explain again how that's going to help it get into a cell in just a few minutes. This is a bacterial virus here called a bacteriophage T4, sometimes just called a phage. This ultimately will land on the surface of a bacterial cell, and these legs here will have little binding sites on the bacterial cell. It will work almost like a syringe, as this body of this virus will kind of work itself down towards the bacterial surface and inject the DNA from inside the head down into the bacteria itself. Viral reproduction works by gaining entry into specific host cells. So the cell that a virus infects, again, is called its host, and it's going to do this through those capsid spikes, those proteins that stick off of the surface or perhaps out of the envelope. They're going to adhere or stick to specific receptor sites, almost like a lock and a key on the host cell surface. So if the receptor here in the silver color is the cell surface that's being infected, the virus particle here has the exact correct key configuration to fit in and be allowed entry into that cell. Here you can see cell A is being infected by virus A. You can see that the fit is very specific. We typically call things that stick off the surface of cells antigens, and they stick into things called receptors on the receiving cell. And you can see that this and this part here fit very nicely together, a lock and key fit. The same thing for virus B and cell B here that it's infecting. Virus B has an antigen that sticks directly into receptor B, a lock and key fit. But if you tried to infect cell A with virus B, you could not do that because this would not fit into this receptor. Same thing with virus A infecting cell B. This antigen would not fit in this receptor here. So most viruses are going to have very narrow host ranges. A lot of times just one species per virus and sometimes even just one tissue. For example, cold viruses will typically hit the tissues of upper respiratory systems and not other bodily systems. AIDS, for example, HIV virus will infect white blood cells, and typically that's what its limit is. It will go after those cells specifically. However, every once in a while, a virus can infect more than one species or tissue. Polioviruses affect nervous tissue and intestinal tissue in people that it infects. West Nile virus can cross species and infect birds, horses, and humans. Let's take a closer look at how a viral infection happens. We'll start with what's known as the lytic cycle. So you can see from our picture here, this is our virus here at the top. Here is its capsid protein coat with the DNA on the inside, and it's going to gain entry through this host cell surface through its lock and key fit. Ultimately, once it's inside, it's going to break apart from right now how it's made. And you can see here the protein comes apart and the genetic material on the inside, the DNA, is now present within the cell. So the viral nucleic acid just entered the cell. That viral DNA or RNA will code for production of protein units in the capsid and to replicate the DNA and RNA sequence. So basically, it's going to build more of itself. So you can see here, this DNA will then go ahead and cause more DNA to be built. Meanwhile, mRNA that's copied from the DNA will cause new protein capsid pieces to be built for ultimately putting together new viral particles. So step number 1, entry and uncoding. You can see it's come apart, uncoded itself. Number 2, we have replication. Number 3, transcription and manufacture of capsid proteins. This is going to rely on the cell's own enzymes, its own ribosomes, and ATP for its own replication. By doing this, it's going to fill up the host cell with new viruses and eventually burst or kill the host cell, releasing new viruses in the body or environment of the original host cell. So by the time these new viruses self-assemble within the host cell, there'll be so many of them inside that often this cell will burst and release all of those new viral particles. Sometimes they'll also be released more slowly through exocytosis. Next, let's take a look at a bacteriophage infection. So viruses can actually infect bacterial cells, and it does so through what we call a bacteriophage virus particle. That was the one that kind of looked like the space probe. So essentially these legs, almost like landing gear, are going to touch down on the bacterial cell surface, find the receptors, bind to them, and then almost like a syringe, it will change its stance, almost bend down and inject the DNA, which is inside the head of the protein coat of this viral particle, directly into the cell of the bacterium. Once inside, the virus takes over the metabolic machinery of that host cell. Looking at that infection as a lytic cycle, which will end with the death of the host cell, it can be subdivided into 5 stages. We have attachment, penetration, biosynthesis, maturation, and release. So step number 1, we have our bacteriophage attached to our bacterium. Number 2, penetration. It injects its own DNA inside of the bacterial cell. The bacterial cell will try to defend itself using what's known as restriction enzymes, which ultimately cut up the DNA inside of a living cell. Step number 3, the biosynthesis of viral genomes and proteins will happen. So ultimately, the ribosomes will read the incoming DNA, change what they do to basically produce more viral particles on the inside of that bacterial cell. Number 4, maturation is basically assembling all of those raw pieces of viral particle into working viruses. Here you can see the phage assembly from head, tail, and tail fibers and how it's put together into a complete viral particle. Step number 5, when the cell can hold no more viral particles, it will then burst, releasing hundreds and thousands of new viral particles. There's also another type of virus that can cause what's known as a lysogenic cycle of infection. For the bacterium, the phage DNA that goes inside will become what's known as a prophage. That DNA will become integrated into the host genome. In other words, it will insert itself into the host DNA. So if you look at the picture here, we can see we have phage DNA, which has injected we have a phage that has injected new DNA inside of the bacterial cell. Bacterial DNA is in the form of a ring, and that DNA has inserted itself inside of that ring and become part of that bacterial DNA. We call that section the prophage. And every time that bacterium will now divide, it will divide with that new piece of DNA being present. So every new cell that comes from that infected cell will be identical and carry that new viral DNA. Now, this DNA can exist quietly, sometimes for a very long time, until specific environmental factors activate the prophage to enter the lytic cycle of biosynthesis, followed by the normal steps of virus action and release when the host cell dies. So ultimately, what's going to happen is it can sit quietly inside of these cells, but if something triggers that viral DNA to become active, it can then switch to the lytic cycle like we just saw, start filling up a cell, and ultimately rupture and release a lot more virus from that infected cell. Let's take a look at it all together. Here we have attachment once again. This is a bacterium, as we can tell by its bacterial DNA and its ring-like shape. We then have penetration, where the phage DNA gets inserted inside of the bacterium. If it goes into a lysogenic cycle, that viral DNA will become integrated as a prophage and attached to that ring of DNA. Every time that bacterial cell reproduces, each daughter cell will now contain the viral DNA. When there is an environmental trigger to activate the DNA that is present in the prophage, biosynthesis will then continue, and again, viral components will be built at that time. During maturation, they'll be assembled, and then when they fill up this bacterial cell, they will be released to infect new cells again. And we're back to step 1. Retroviruses are special types of viruses that have RNA instead of DNA. But what they're going to do is they're going to go ahead and use an enzyme called reverse transcriptase that copies RNA into double-stranded DNA. Now, the central dogma of biology typically goes the other way. DNA is copied into RNA, which gets translated into proteins. But these animal viruses of RNA have the ability to basically be translated into new DNA, which will now stay with the cell by help from an enzyme called reverse transcriptase. And that's how the HIV virus works. Let's take a look at how a cell gets infected with a retrovirus. Here's your viral particle with reverse transcriptase on the inside, and this genetic content on the inside is RNA. As it enters the cell, the RNA gets released. That RNA will be copied into DNA using reverse transcriptase as an enzyme. The DNA will then insert itself into the host DNA as a provirus. Now, we have combined DNA from the original host and the virus, and mRNA's job is to copy segments of DNA. So it will copy both the original host and the newly integrated viral DNA to produce new virus parts, again, allow for assembly of new viral particles, and then allow them to release from the cell and infect new cells. With HIV, white blood cells are the target. They're going to be infected. The viral DNA will enter the nucleus of the cell, create new virus, and exocytose the new virus particles outside, like we saw in the diagram. It is a steady, slow infection of the white blood cell. So, what is their function? Why does it target a white blood cell? Well, that is its target. We said often these viruses are tissue specific. And the white blood cell's function is an immunity. So if you slowly have your white blood cells being negatively affected by a virus, the organism's immune system will then be compromised, and they'll be susceptible to infections from things that normally the body can defeat. Viroids are extremely small, uncapsulated, circular RNA strands. They induce the cell to produce more viroids. So these pieces of genetic material are floating around and have the ability to get inside of the cell to produce more of themselves. We see a lot of these in the plant kingdom. Prions, or proteinaceous infectious particles, come from misshapen proteins that induce normal proteins to change their shape in a biochemical response to contact. So if we look at our little diagram here we have a normal protein, and this protein is called a prion. All the prion has to do is come into contact with that normal protein and change it into another prion, which will trigger a chain reaction every time a new protein is touched, forming aggregates of prions. We see this in mad cow disease and Creutzfeldt-Jakob disease. We've reached the end of our virus lecture for today. Remember that viruses are acellular collections of biomolecules that infect host cells. Always reflect back on your learning targets. Let's take a quick look once again. Number 1, be able to explain whether a virus is technically alive or not. 2, can you describe the structure of a virus? 3, explain how a virus infects a cell. 4, compare and contrast lytic versus lysogenic viral cycles. 5, explain how retroviruses work. And 6, define viroid and prion. More to come on viruses. I'll see you next time.